Hello, everyone, and welcome to today's webinar. And my name is Jessica Duggan, and I'm the parent of a little girl with SYNGAP1 disorder. We're very excited to continue the SRF Research Fund webinar series. The goal of the series is to empower your communications with clinicians as you get more clear knowledge of SYNGAP. We also want to give a plug for our next presentation, which is interpretation of variants in SYNGAP1, and that will take place on August 19th at 10 a.m. Pacific time with Dr. Eduardo Perez Palma. Our talk today is improving the assessment of SYNGAP1 and related genetic conditions by creating online measures for parents and patients. And I have the great pleasure today to introduce today's speaker, Dr. Thomas Frazier. As a clinical psychologist, Dr. Frazier has focused his career on improving autism identification, developing and studying new treatment approaches, creating and refining measures for tracking autism symptoms and the quality of life in individuals and families affected by autism. He was the director of the Cleveland Clinic Center for Autism and the chief science officer at Autism Speaks. Currently, he's a professor of psychology at John Carroll University. Today, Dr. Frazier will introduce us to a study which is trying to develop and validate an online neurobehavioral evaluation tool on SYNGAP1 and other neurodevelopmental genetic syndromes. This study is in part possible with a grant funded by SRF. And I want to remind you, a recorded version of this webinar will be available on the SRF website under webinars. By the end of the presentation, you'll have the opportunity to get answers to your questions, and we'd love to hear from you. So please write your questions in the Q&A. And for those of you who are just joining us, welcome again. And our speaker is Dr. Thomas Frazier, and his talk is Improving the Assessment of SYNGAP1 and Related Genetic Conditions by Creating Online Measures for Parents and Patients. And it's now my pleasure to turn things over to Dr. Frazier. Thank you.
Thanks, Jessica. Appreciate it. Is it okay if I share my screen now?
Yes, please.
Great. Thank you for that great introduction. Really appreciate it. As Jessica mentioned, over the last really now 12 years, 13 years, I've been interested in genetic syndromes associated with neurodevelopmental disorders, including autism and ADHD, but really more broadly, genetic syndromes that influence neurodevelopment. And a little bit before the pandemic, I started to study different methods that we could use to improve the assessment of genetic syndromes and neurodevelopmental disorders. And so this project that I'm going to describe to you today is really about how we can do a better job of making sure that we have the right assessment tools, that those tools are easier to use for patients and informants and parents and family members, and that ultimately those tools help us to track change and outcomes in patients, so that when we institute any kind of intervention, or if we just are following patients over time, we can better understand what's happening with them and how they're improving. So this project is funded by several different organizations, but I just want to call out SRF, SYNGAP Research Fund, for its funding for this project. It's really fantastic. When I first contacted Mike regularly, he just really understood what I was trying to accomplish and how it would benefit SYNGAP1 patients and families. And so I'm very grateful for this support. And I hope you'll also see that what we're doing can be useful for the community. We're developing something called a neurobehavioral evaluation tool, or a NET for short. And obviously a reasonable question to ask me is, what is a NET? And you know, the first place to start in answering that question is, what is neurobehavior? So neurobehavior really is just a fancy word that represents all of the different brain processes and brain functions that help us to thrive in our world. And there are many different examples of these, but I'll just give a few positive and a few negative examples. So positive examples would be like attention. We allocate our attention to things that we want to understand, that we want to process, that we want to take in, that we want to remember. And so attention is really a very, very important cognitive function, because it allows us to understand our world. Another example is executive function. You might have heard of executive functioning or executive functions. This includes a whole bunch of different cognitive processes, but the most common ones are things like planning, organizing, inhibiting, sequencing. These are all the kinds of things we have to do to be successful, because if we don't plan things, we can't figure out what we need to do with our day. We can't manage our behavior. If we can't inhibit our impulses, we might do something that we regret later or that isn't productive. So these are just some examples of different aspects of neurobehavior that we're going to want to measure with our net. And then dysfunctional examples or negative examples would be things like anxiety, right? If we aren't doing a good job of managing our emotions or regulating our affect, we can sometimes experience anxiety or extreme fear. Another example would be challenging behavior. Challenging behavior is very common in individuals with neurodevelopmental disorders. It can range from things like oppositional behavior to aggression, self-injury, elopement, property destruction. And certainly not everybody with neurodevelopmental disorder has challenging behavior, but when folks do have challenging behavior, it's really important to measure it and track it, because you obviously want to make a difference there. And challenging behavior is the #1 reducer of quality of life in families. So we're building an evaluation tool to assess these different brain processes. And we had a number of different motivations for this. You know, somebody who's involved in this field might look at me and say, well, Dr. Frazier, I appreciate your effort here, but we have a lot of existing tools here. Why do we need a new one? Well, first of all, the existing tools are not specifically designed for neurodevelopmental genetic syndromes like SYNGAP1, like P10, like ADNP, like some of the other neurodevelopmental genetic syndromes that we're studying. And so we need to actually make measures that are more appropriate for these patient populations. And to do that, we're including clinician scientist experts, caregivers, and patients in the development process. We also know that many of the items on existing tools are kind of irrelevant, or maybe not the best way to phrase or frame something for the cognitively impaired or nonverbal or minimally verbal patients that are often a part of these communities. And so I had a lot of parents that would come up to me, for example, after we have them fill out these existing tools, and would be like, well, I only filled out about half of it. And you know, when I would talk to them about it, I realized, well, they filled out half of it because the other half of it wasn't really relevant to their child or their situation. So we have to be much more careful about that when we develop tools. We need to collect data repeatedly as well, right? We need to be able to do this in a way that is easy as possible on the patient and the family. And so this is even before the pandemic, I realized, you know, we need to do this online and at home as much as possible. So our measures are going to be online measures that can be collected from home, really in the safety and leisure of the home environment, make it easier on patients and families. And then we want to have a bunch of different measurements here, right? So we need to measure all kinds of different aspects of neurobehavior, and we need to have those measurements be normed at the same time so that we can get a really accurate profile of the patient's strengths and weaknesses. We can compare that profile not only within that patient over time, but also to other patients in that community. If we institute an intervention, we of course want to be able to track that over time. And there are two basic ways we're thinking about in terms of our project to collect neurobehavior. The first one is kind of obvious. It's a survey or set of questionnaire scales, right? And for this one, we'll be asking a parent or another informant to fill out these surveys that ask about the child's strengths and weaknesses, behavior problems, daily living skills, etc. And surveys are really important because obviously the informant or the parent or the other caregiver that fills it out oftentimes has a lot of information about the patient's behavior. And so we need that sort of really detailed look at the patient's behavior. But we also want to collect data directly from the patient, and we can do that now using webcams. Most people, not everybody, unfortunately, but most people have a webcam. And for this study, even if you don't have a webcam, but you have a computer, we'll send you a webcam. We can also use tablets for this study as well, because tablets have a webcam. But webcams allow us to collect data directly from the patient. And I'll show you just now sort of, this is a little bit of a busy slide, but I'll just sort of walk you through some of the different ways we can collect this kind of data. So Webcam measures allow us to get a video of the patient as they're watching the screen. And from that video, we can do two big things. One is we can track where the patient is looking on the screen. And two is we can track their facial expressions, their reaction, their engagement with the screen. And so using those measures and changing what we show on the screen, we can collect information about different cognitive and emotional processes. And we do this primarily by showing static slides, or more often we show videos. And those videos are pulling for different kinds of neurobehavioral processes or cognitive processes. So for example, on the top left here, you can see we show a slide that has different pictures on it. And what we'll do is when we show those pictures, we'll say, look at the person kicking. And that's an example of a slide that's pulling for receptive vocabulary, receptive language. And by tracking where the person looks after we give the direction, then we can see that we can look at their receptive vocabulary. We also do things like just showing people's faces and asking people to look at certain faces, like look at the happy face. And that on the top right here, you can see that we track where people look. And because of that, we can track to see if they're looking at the happy face. But even then, even if they're not looking at the happy face, we can also track whether or not their facial expressions have changed, right? Are they showing a more positive or a more negative facial expression? On the bottom left, you can see we show some stimuli that have like a social half and then a non-social or an abstract half. And on those stimuli, we can track social attention because we all have this general preference for looking at social information, but that preference differs across people and over time, and we can measure that preference and track it over time. And then lastly, this isn't the only, these aren't the only stimuli we show, but I'm just giving you examples. The last example here is a joke stimulus where a young child is telling a joke, just a funny sort of dad joke. And we can again track where they look in their facial expressions to these things. So these are just some examples of different stimuli we can use to better understand cognitive functions using the webcam. So in terms of the specific goals of the study, we're going to use a data collection here from SYNGAP1 patients as well as other patients and controls. And we're going to use that data collection to evaluate the ability of our survey measures and our webcam measures to track changes in behavior and functioning over time. And so I'll describe a little bit more how we're going to do that, but that's the main goal of the study. And by doing that, we're going to be able to validate these measures for people with SYNGAP1 and other genetic syndromes, as well as people with neurodevelopmental conditions that don't have an identified genetic syndrome. So for this study, we'll be recruiting patients with SYNGAP1, obviously, that's why we're doing the webinar. We'll also recruit patients with PTEN mutations, ADNP mutations, SCN2A, STXBP1, NFIX or Milan syndrome, and also autism spectrum disorder. And we'll also have healthy controls, including sibling controls, which we're always looking for, and unrelated healthy controls as well. And the controls are just to help us to understand how the measures are functioning in people that don't have a genetic syndrome or a neurodevelopmental condition. In terms of eligibility for this study, our primary participants, the patients, will be ages 3 to 45. Unfortunately, we can't go above 45 right now, and I know that's been frustrating for some families, for some patients. But for right now, the FDA kind of wants us to keep it to a narrower age range, but it's still a pretty broad age range. They have to be diagnosed with a genetic syndrome or autism spectrum disorder. They have to have a willing supporting participant, like a parent or another family member or some kind of close informant that knows their behavior. And they have to be willing to complete or at least attempt to complete the webcam measure. The supporting participant must be an individual who is knowledgeable of the primary participant's behavior and functioning, and they're going to be the person that completes the survey measures. Both participants have to have a reliable internet connection, and they have access to a tablet with a stand or a laptop or a computer. Like I said, we can provide a webcam if the webcam is broke or not working or just don't have a webcam, but we need to have that computer. So for the survey portion, The supporting participants are going to complete a survey that helps measure behavior and functioning of the primary participant. And they're going to be asked to complete that survey at 3 different time points: the initial or baseline, 1 month later, and then 4 months after baseline. So 3 months after the first follow-up, but 4 months after baseline. The survey links will be sent by the research coordinator, who happens to be on this webinar. Her name's Katie Huba, and she'll send the links by email to make it easy on people to complete. And we estimate that each survey will take about 20 to 30 minutes to complete, and the webcam measures can take anywhere from sort of 15 minutes to 20 minutes to complete. So for the webcam portion, the primary participants are going to be asked to watch several videos. The videos, like I showed you earlier, are going to measure different cognitive functions or thinking skills like language, attention, processing speed, or the time it takes to complete a task, things like word recognition. The videos do not require a verbal or a physical response. That's intended to make it easier, because some patients are nonverbal or minimally verbal or non-speaking, I should say, and or may not have the ability to generate a physical response. The videos are going to be approximately 60 seconds long. We try to keep the videos pretty short, because before each video we have to calibrate gaze, and we don't want the videos to be too long because we might lose the person's gaze, and also people lose attention, and we wanted to keep these videos short so even young kids or people that have trouble paying attention might be able to do the videos. So again, the same 3 time points are going to be used for the webcam as for the survey measures, and baseline, 1 month follow-up, 4 month follow-up. And again, our research coordinator, Katie Huba, will send the links to the videos via email. And like I said, the webcam measures take 15 to 20 minutes to complete. You can do the videos in chunks. That's really important, because some people might say, hey, we can't, my son or daughter are not going to be able to do these videos in 15 minutes. They're just not going to be able to keep their attention. Well, guess what? You can do each of the videos in really short chunks. So you can do 1 or 2 or 3 minutes and then take a break and come back to it later and do the next 2 or 3 minutes until you're all the way through the video. So we're trying to make it as easy as possible on folks to do these. We will be compensating participants. There's a $30 Amazon gift card per survey completed. And then there's a $30 Amazon gift card per webcam measurement completed. So across the whole study, because we have 3 time points, it's possible that people will earn a total of $180 in Amazon gift cards. Even though it's not a long study, we do know that there's, people have to take their time, their energy, to do this, and the supporting participants are oftentimes going to have to help the primary participant do the webcam measures. So we just wanted to make sure we're appropriately compensating people for their time. If you feel like this might be a challenge for you to participate, the research coordinator is available to assist you. They can jump on a Zoom or a Google Hangout and try to help you through a part of the process, or give you tips and tricks for how to make this easier on the primary participant. You can always withdraw from the study at any time. There is a third opportunity for participation, and that's if siblings would like to participate. Remember, I mentioned that we would like healthy control siblings, ages 3 to 45, of the primary participant that do not have a genetic syndrome or neurodevelopmental condition could also participate. We just want to make sure you're aware, if they do participate, then the supporting participant will have to fill out surveys for them as well. So they'll be doing 2 sets of surveys, one for the primary participant and one for the sibling. I did want to sort of end on just, I went through a lot of the logistics, and thanks for bearing with me, but why does this really matter for SYNGAP1 or for other genetic syndromes? And, the big deal here is that we have an opportunity to improve clinical practice. And these new net measures, if we develop them and validate them in the way that we hope to, could really improve clinical practice by making it easier on the clinician and the patient and family, by having some measurements that are collected ahead of time that can track function and behavior. And the clinician can just use those. You don't have to fill them out in the waiting area. You don't have to fill out a bunch of paper forms, which can be quite tedious to fill out. Trust me, I've done it for my son. many times. So this is a way for us to improve clinical practice. Our new measures here, the NET, could also be used to track progress in future longitudinal studies and natural history studies. We need to learn a lot more about SYNGAP1 in order to be able to come up with the best ways to help patients. And so these kinds of longitudinal studies can be really important. And then lastly, and probably the thing that's oftentimes most on people's minds is, are there going to be future clinical trials? And I hope the answer, and I'm pretty sure the answer to that is going to be yes, there are. But the NET is a way for us to improve and have better outcome measures for those clinical trials. One of the reasons why some clinical trials in the past have not been successful, we think, is because the measures themselves were not appropriately capturing change. And so these new NET measures we think are going to be much more effective and useful for future clinical trials. This is the investigative team. Obviously, myself and Katie Huba I've introduced. We also have Merkel Ojerovic, who is a senior research fellow at the University of Melbourne. He's a psychiatrist. He's been helping us with developing the measures. And then Antonio Harden is a child and adolescent psychiatrist at Stanford University, and he's the director of the Division of Child and Adolescent Psychiatry there. Longstanding interest in neurodevelopmental disorders, and he is also closely involved in helping us with the project. And then we have several teams of experts that are also engaged with us on developing the measures. So I'll end here and just, I'd love to take any questions that people have or any issues, comments, concerns that people have. We're really hopeful that the project is going to make a difference. And, you know, but we need your support. We need your help. We need you to participate. We think we're trying to make the participation as easy as possible on you, but we do know that it does require time and effort. So thank you for considering this. Thank you for watching our talk. And it's usually more fun to do questions if people have them anyway. So I'd love to stop here if that's okay.
Thank you so much, Dr. Frazier. That's amazing. So I'm looking in the Q&A and in the chat, and I'm not seeing anything yet. So I'll just start off. Hopefully people will write in some things. Well, actually, yeah, already there is some. So Genevieve writes, Dr. Frazier, thank you for the interesting talk. For the survey, because it's filled out by the primary participant, is there age limit? So I think that might be, is there an age limit?
Sorry, I wasn't clear on that. The primary participant is the patient with a neurodevelopmental disorder or genetic syndrome. So the SYNGAP1 patient in this case is the primary participant, and there is an age limit on that. That's 3 to 45. But for the supporting participant, the person who would actually fill out the survey, there is no age limit for that person. So as long as they have a really good understanding of the primary behavior, as long as they really understand that person, that person with SYNGAP1, then there is no age limit for them. Obviously, most of these folks are going to be parents, oftentimes moms who fill out the surveys, but we don't have an age limit.
That's great, thank you. So I had a couple questions as you were going through. One is, they're pretty specific to the setup. So would the tablet or computer with the webcam need to be on a stand? You know, like I know my son sits on his bed and watches his iPad, looking kind of down on it, sitting on the bed. So I'm kind of wondering what the physics of everything is for this.
Good question. Yeah, the webcam has to be stable. So we can't have the child holding the tablet because obviously that'll move a lot. So we need to have the tablet like on a stand or sitting up in a stable position. And then the calibration process allows the supporting participant and the primary participant to sort of see where their head needs to be. Luckily, there's some wiggle room there. Like they don't have to stay perfectly straight the whole time. They can move around. Their eyes can even go off screen for a little bit, and we can usually recapture them back. But the tablet itself needs to stay stable. We usually, if you have an option between a tablet and a webcam, we usually prefer the webcam in general. I'll say in general, because generally speaking, those are going to stay more stable. regardless of the situation. And oftentimes the screens are a little larger, because the larger the screen, the more accurate the gaze capture is. But Katie can tell you, we recently had a patient, we've been doing a lot of initial testing on this, and we recently had a patient completed on a 10-inch or maybe slightly smaller than 10-inch tablet, and it worked. So we recommend tablets that are larger in size than that, but we were pretty impressed that we actually got pretty good data from a smaller tablet. We don't want people to use smaller tablets unless they absolutely have to, but we recommend a larger tablet size.
Okay, that's great. It sounds like maybe at least the setup I'm thinking of might be a laptop might be best, as it has its own kind of sort of a stiff laptop would be great. Okay, so I have two more questions. So you were saying that you can ask the participant to look at various things. Are these auditory commands embedded in the video? I know, like for my child, he doesn't get auditory commands that well, but he does paired with pictures. So I'm wondering if I'm just wondering what the commands are to look, and what if you've kind of thought about that kind of approach.
Totally, yeah. You know, we know that a fair number of the patients that we're trying to evaluate are going to have some receptive language challenges or some difficulties with following particular directives. So we have 4 basic paradigms, and I'll just sort of walk you through them since you asked about. One is a social attention paradigm that does not have many, it has a few, but not many specific directives in it, okay? The only specific directives in that paradigm are look at the happy face, look at the sad face, look at the mad face. Otherwise, that paradigm, we don't really have directives there. The second paradigm is a receptive language paradigm. For that, we do have auditory directives. Look at the person kicking, look at the ball, look at the person drinking, et cetera, et cetera. We know that there are some individuals that will struggle with that. In our experience, so we actually already have a published study on that paradigm. And that paradigm, we were able to get about 90 to 95% of individuals, including some very significantly impaired individuals who are 2 and 3 years old, to complete the paradigm. But that doesn't mean everybody can complete it. And there are certainly some kids that just can't follow the directives. The good news here is we ask you to try the first 1-minute video. And if it really looks like to you that the child or whoever is attempting to do the task, if that person is not able to do the task, you do not have to persist through the remaining videos. So for example, if it's obvious to you that this is just not a paradigm that works for your child, then it's perfectly fine to actually skip those remaining videos for that paradigm and go on to the next paradigm. Because we don't want to torture people. Like, we want to make it, really difficult for people. So the third paradigm we have is a single word reading paradigm. The Simons Foundation asked us to add that paradigm, so we're going to put that one last, because we know that there's a fair number of individuals that don't read. And so we understand that, we get it. It was a request from a funder. We're going to put that at the end. If people don't want to even try that, that's okay by us. It's fine. And then the third paradigm, I think almost everyone can, the fourth paradigm, which we're going to put third in order, is I think almost everyone can do it. It's meant to just be a way to track the person's gaze on the screen. There is a directive, but the directive is not too bad, which is sort of just look at a particular object. So like, look at this object. And even if the child doesn't do well with auditory, the supporting participant can point at the object on the initial screen and say, look at that object, right? And so there's a practice slide ahead of time, and we're sort of orienting the person to look at this particular thing. So let's say it's a red balloon. They will look at the screen and we'll show them, this is a red balloon. Look at all the red balloons. And then on the next screen, there'll be red balloons mixed with other things. And we just want the person to look at the red balloons. So we think on those paradigms, we can get a pretty good, maybe not 100%, but close to 100% of people will be able to do those tasks. Does that answer your question, or?
Yeah, it mostly does. I'm gonna just dive just a tiny bit deeper, because I think there's one nuance that I didn't get across in my question, which is it's about a difference between taking in directions auditorily versus visually. So if you wanted someone to look at someone kicking a ball, you could do like a quick video of a ball being kicked and then go to a still of two things. And one was a foot about to kick a ball and one was like a swimmer, right? And then they might be able to like look at the foot about to kick the ball, sort of showing that they understood that was what it was. It's that kind of thing, just sort of visual versus auditory instruction without reading, you know, although I will say that I think for SYNGAP, I think kids have a very good visual memory, and I think sight words are very common for SYNGAP kids.
That's something we'll definitely consider. I think one of the trade-offs we're trying to deal with in this paradigm is we only have 60 seconds to capture gaze before we have to go on, and we need a certain number of items in order to make useful tasks. So we have to be very careful about how much practice we can do ahead of time, but that's definitely a cool idea. And I think maybe we could even start, Katie and I can start thinking about how we can make versions of that to help certain participants be able to understand those skills. I think that's a cool idea. So definitely, Katie, I know Katie's taking notes, so let's we'll get back together on that and see if there's a way for us to implement something like that.
Yeah. Okay, cool. So there's another, there are a few more questions here in the Q&A. Same person before, Dr. Frazier. Do you take into account any various treatments for the genetic syndrome or neurodevelopmental disorder that participants may have, and how they will influence the results from the survey and webcam?
Yes. Before each survey, we ask the supporting participants to complete a questionnaire that asks about treatments and any other environmental changes that the patient is experiencing. And we ask for a little bit of detail around that. So yes, in our analyses, we'll be able to look at whether or not and the treatments are impacting the measurements that we're taking.
So yeah, and I guess a follow-up of this is, I think that what you said at the outset is this initial study is to look at these measures. Like the study is to look at the measures, not to look at the participants really, in some sense.
Yes. Yeah, we want to understand what the participants look like on the measures, but you're right. The main thrust of the study is really to develop measures that are useful for the SYNGAP1 patient population and other neurodevelopmental or neurogenetic conditions. And so we're not so much focused on saying, hey, does this treatment work? We won't be able to answer those kinds of questions. What we will be able to say at the end of this, hopefully, is these measures look appropriate and useful for the vast majority of the SYNGAP1 population. That's what we're hoping for.
Yeah, that is, that'll be such a great day. Okay, so in the chat from Jess, a few questions here. So is the video content varied or the same over the evaluation sessions?
Yeah, so the video content for the webcam will be the same across the evaluation sessions. And the reason for that is there's some technical reasons for that, but we do know that people might get annoyed or bored with it. But remember, we're putting 1 month and then 3 months in between administrations. And so in general, when we've done this in the past, we don't see major differences as a result of repeating the information. And we have ways of adjusting for that when we do see them. So we are going to show the same content over time. The other nice thing about not varying the content or the paradigms too much is that we do want to see how much people might improve just due to practice effects, because that'll give us a sense in the future of whether or not it's useful to actually do sort of an initial practice before, let's say we were to implement these measures in a clinical trial. Then if we learn that practice really mattered, then we actually want to administer the measures a couple of times before the treatment started in order to make sure that we were measuring change and not just practice on the measures. So we're gonna use the same kinds of video content over time. Some people have told us after the third or fourth time they see it, like, yeah, I was ready for the, you know, whatever that particular stimulus was, I was ready for that. But it turns out that it doesn't change their gaze a whole lot, and it doesn't really change their responses overall a whole lot, so far anyway.
Thank you. And I've been a poor host to not ask you to stop sharing your screen so we can all see each other.
Oh, yeah, sorry.
Okay, and then Jess also asked, can you speak about how you would expect improvements to look over time?
Yeah, so we're measuring different processes here, different thinking skills, and other kinds of problems that people might experience. And so if we have a good measurement at time 1, and we repeat that measurement, say a month later or 3 months later, then what we can do is we can sort of see how much the score on each of these measures changed. And if the measures are reliable, and that's our goal, is to develop really reliable, useful measures. If the measures are reliable, then hopefully even small changes in the patient will show up on the measures, whether they're the survey measures or the webcam measures. They'll show up. And what I mean by that is the score will change over time, and that score will show us what improvements people are experiencing. So for example, in general, people with any kind of neurological condition, if they improve with a treatment, one of the first things that improves is their attention. So this is true in many, many different neurologic problems, neurogenetic conditions, etc. And so by measuring attention, and by the way, we're measuring attention in 2 different ways. We're going to ask the parent, and we're going to measure attention through the webcam. By measuring attention, we should be able to see if there's any improvement from time 1 to time 2 to time 3. Does that address the question, or am I missing it?
Yeah, that does. Thank you.
I guess a follow-up to that question will be, how about kind of variation? You know, how do you think this will, I guess within each trial, if you have 15 to 20 minutes, you'll see some variation in that time. Is that the main way you're going to look at variation, or?
Oh, you're saying variation within the short time period that we collect the measures?
Yeah, well, let's see. I guess I'm just trying to imagine what the data looks like. I'm imagining graphs and standard deviations and points and p-values, and I don't quite understand.
Yeah, so for each measurement, let's just take one of the measures we'll collect from the webcam is a measure of sustained attention. How long can the person keep their attention to the screen? It's a very basic measure, right? Very simple measure. And it really is just going to have to do with how long was their gaze detected on the screen, okay, across all of the different things that they tried to look at. And it comes as a proportion. It turns out it's from 0 to 100. 100 means you looked all the time, and 0 means you didn't look at all, right? And so we'll use that measure to compare that individual over time and also across patients, right? So different SYNGAP1 patients, so we can understand things like, if you're a SYNGAP1 patient that has a particular other problem, do we see that also manifest in sustained attention or not, right? And so we'll be able to look at those measures, each individual measure, and sort of compare them within each patient and across patients. And then, of course, if we implement these in clinical trials, then we'll be able to see, is the treatment really improving this person's sustained attention? And we're going to collect, the webcam itself will probably allow us to collect at least 15 to 20 of these types of measures. And then the survey measures will also collect roughly 10 to 15 different scores.
Okay, that's great.
It gets complicated as you get into all of it, but
yeah, of course. Well, and I'm also thinking, you're talking about 0 to 100, and I'm assuming that typical cognition would not be 100.
No, typical
Right.
is
So
low 90s. Yeah.
yeah, so there's going to be some kind of, there could be staring that isn't engagement, or, anyway,
Yep, totally.
it's a very interesting topic you have here. Okay, we have a question in the Q&A from Nancy. Hi, thank you so much for developing these measures. My question is about assessor directions and prompts. How will this influence these results? Will the family members receive directions and/or practice prior to administering?
Yes, absolutely. Nancy, great question. Thank you. I didn't pay you to ask the question, but this is a really great question, because the answer to all of these is yes. We will be giving you directions. We will be showing you. We have both videos, and Katie can actually jump on a call with you to walk you through how to do this. The assessor directions and prompts could influence the results, but what we're trying to do is we're trying to get a best performance here, right? We don't want to hamstring the person with SYNGAP1 by saying, hey, you figure it out. We actually want to get that person to the optimal performance possible for them. And so we want the supporting participant to help them as much as possible without giving away answers or telling them where to look, right? So we're going to actually, Katie, like I said, Katie, and we have some video methods, and we also have a lot of instructions for you. We're going to be trying to train you up on the best way to get the best performance possible from the person that we're administering the task to, without cheating, right? without actually doing it for them. And so there's all kinds of different ways we can do that, including practice administrations, including having Katie give you tips and techniques and walk you through it. And we have a lot of different ways to help you with that. So as long as you're willing to work with us on it, we're totally up for working with you on it. And in fact, we expect that probably somewhere around 20% of individuals will not be able to validly complete some of the videos the first time through. And that's okay, because we're willing to work with you to actually try it again. If you're willing to work with us, we'll try it again. And in fact, in our pilot testing, what we've seen is for some individuals, they'll do it the first time and it's sort of borderline, maybe it's not really valid. And then we go back and we try it again and it looks great. And we want that optimal performance. We're not trying to hamstring, people here. We want them to do the best they possibly can.
Great, thank you. Well, I don't see any more questions right now in the chat or the Q&A, but I have kind of a question. It's not really fair to you, but what I was wondering about even sort of broader measures. When I first was thinking webcam, I was thinking, you could have something overnight and sort of show nighttime behaviors, or you could have like much more constant, something more like a wearable where you're really constantly collecting data. So it surprised me. I'm excited to hear what you're doing, and this sounds amazing, so I don't want to detract anything from it, but something that was a little bit surprising to me was that these are 3 fairly short moments of time over a few months, and I think you're going to get great data from it. But I'm always wondering, how can we tell what's happening with our kids, especially when they can be so angelic and then they can be so unhappy.
Yeah. Well, I have a good friend named Matt Goodwin who's working a lot on wearables for kids with neurodevelopmental syndromes. And I think wearables are a fantastic other way that we can do a lot of this stuff. And even like webcam behavioral observation stuff is really cool as well. There's some tricks in that right now. There's a lot of work that needs to be done in AI and machine learning to sort of make sure we can extract that data efficiently. But Matt Goodwin's doing really great stuff with wearables like watches that you can wear on the wrist or even the ankle. And you can get really good autonomic data. We were more going for cognitive data, like thinking processes, but autonomic data you can get from the wearables. And actually, I'm working with the company to try to get them to collect some data on the pupil as well. we're not going to be doing this the first time through, but for those of you that aren't aware, like the pupil is a really interesting window into our autonomic nervous system. And you can learn a lot about sympathetic activation and also sort of parasympathetic calming of our autonomic nervous system through the pupil. And so we would ultimately also like to collect pupil data. We're not going to have that the first time because there's a lot of tricks in how to do that. But that's another way we can gather information. But I totally agree. There's all kinds of these different opportunities out there right now. We're only taking advantage of one small corner of this space, but we would love to include other stuff in the future.
That's really great. I just want to ask, do any of the other panelists have questions, or does Katie have anything she wants to add? Because I'd love to hear from Katie if she had something. Or it looks like Hans is unmuting. Let's hear Hans.
Yeah, thank you, Catherine. Thanks everybody and Dr. Frazier for the webinar. In the past, when I've done surveys with my son, I've developed a degree of frustration because he's nonverbal. And a lot of times the question and answer component of the survey is frustrating to try to, find the right answer. And there is some significant heterogeneity to the subjects you're looking at between P10 and ADNP and SYNGAP, STX, and so forth. Can you expand on that, please? And do you stratify your analysis based on verbal or nonverbal? Is it an adaptive survey?
Yeah, so we aren't able to start with an adaptive survey, but you've anticipated we do want to move to adaptive surveys in the future. So we're starting with a more traditional looking survey, but as we gather more and more data, we think it can become more adaptive. To your verbal nonverbal point, my son is also nonverbal, and I'm very sensitive to this issue because I've had the exact same experience that you just described. And I can't say that every item we've developed is going to make you excited about the survey, but I can tell you that through my own experience and through a lot of our experts, we are eliminating many of the traditional verbal items that you would see on these measures. And so what we're doing is we're focusing on other ways to get at the same constructs without asking verbal questions that involve a verbal component to it. We're also going to be offering people the ability to choose a response. So we want people to sort of take a guess, but then we're also going to be offering people for each item the option to say, I wasn't sure, or I didn't think this item was super applicable to my child. And so hopefully that gets rid of some of the frustration that people experience. Like I said, I don't think we're going to solve that problem entirely, but I am very sensitive to it. So by the way, we will collect whether or not people are verbal or nonverbal, and we will also collect some other clinical information that will help us to stratify the analyses. Yes.
And then I was going to ask, in terms of, it's great to hear about gaze tracking and attention. If there's a tennis racket on the screen, you're unlikely to get attention from my little boy. If there's a garbage truck, he's going to lock eyes with that garbage truck and not blink. So is there an ability to personalize what will provoke attention in the subject?
We don't have the ability to personalize the provoking of attention, but we do have on the back end, on the analytics side, we have the ability to look and see, okay, if we presented something that the person was really engaged with, maybe it's just by happenstance we presented it. Then for that stimuli, what does their cognitive processing look like there versus one where they were not as interested, not as engaged? So we do have the ability on the back end to look at that. And that is something I'm also pretty sensitive to because my son has a similar, not garbage trucks for him, but he has similar things that interest him visually. Unfortunately, on the front end, we have to be more standardized with the presentation. And so we can't assess what's interesting to them and then put those into the task. We thought about that initially, but it became it becomes too challenging to make sure that the difficulty is relatively matched, if that makes sense.
And then one last question. Has the FDA shared any insights into their thinking in terms of maybe the Angelman's ASO pre-IND meeting or any guidance documentation that's come out?
The FDA, as I'm sure you probably know, has their patient-focused drug development processes that they've put in place. And through the P10 Research Foundation, we are engaging the FDA on these measures. And so we've already had one preliminary meeting as we're developing the measures, and the FDA gave us input in that meeting, including adding some additional steps for us to go through in the measure development process. Once we're done with that initial measure development process, the FDA has asked us to come back and have another meeting. And so we'll have another meeting probably somewhere in the September, October timeframe, at which point we'll be asking for their blessing on moving forward with what we're calling phase 2, but for this group is really the major data collection. We'll be moving forward as soon as the FDA gives us their blessing on the measure development process. And then, of course, after we've collected the major set of data, we'll be bringing it back to the FDA, again, for their review of the data, what things look like in terms of reliability, validity, and we'll be asking for their blessing then for the measures for future work.
That's great. Thanks so much.
I did miss one question from Jess. How are you recruiting for this study for people with ASD?
We have some local clinics, and then I obviously have a connection to Autism Speaks, so we're probably going to do some national recruitment through them. But we're always open for referrals, right? So if there's just a family that participates that is associated with SYNGAP1, and they know a family that has idiopathic ASD, we'd be happy to have them referred to the study as well, as long as they meet inclusion-exclusion criteria.
Great, great, thank you. And Katie, did you want to say anything?
Hi, everyone. Yeah, I just wanted to say hello. And as Dr. Frazier mentioned, I will be here to help you and assist you in any way possible to make this as easy as possible for you guys during the surveys and the performance measures, and how we can work together over Zoom or Google Meets to, make it as easy as possible to get as best data as we can. So, and just from some things that we've seen so far, we've been able to get some really great with just results, even with some patients who parents thought it wasn't going to be a possibility to gather that. We've been able to do that, and that's just with a small pool that we've been working with so far. So I'm really hopeful and excited to work with you guys during this project, so I look forward to it.
Thank you. And then before I end, I just want to say thank you so much, Dr. Frazier and Katie and your whole team. This is such an essential, crucial point that we need measures, good measures. And you've clearly been thinking very deeply about this, and we just, couldn't ask for anything more. So that's it for me. And I don't know if Marta or Jess will.
I just want to give you thank you. And this work just can change life for our kids in the schools. I still remember when my daughter, when she was little, getting evaluated in the school, and she couldn't do anything. And then there was no measure possible because she was not verbal. And we know a lot of these kids, the expressive understanding is zero, but they cannot express, but they understand a lot. Then this is great for the future of all these kids with these disabilities of being unable to express themselves. Thank you so much.
Thank you for having me. And I just do want to put in a plug that we do expect to start recruitment in a month or so, but we'll obviously work through the foundation at that point and let folks know when we're ready. But just thanks for everybody who watched this, and appreciate all your time, and appreciate the foundation's desire to do this with us, and just thanks so much.
Oh, Dr. Frazier, if you can mention the international recruitment, we were talking informally initially, but I think that's very important for.
Yeah, we're also able to take, so one of our inclusion criteria is just that people have to be fluent English speakers. That is unfortunately, that is a limitation. But as long as they're fluent English speakers, we can include people internationally. We've already had interest from folks in Australia, Canada, the UK. We've had folks across other parts of Europe that are fluent English speakers that have also, wanted to participate. So we definitely encourage international participation. We're working with several people in Europe and Australia. I have connections in Canada as well. So, definitely we're open, we're excited. We want people that are excited about participating. We need that kind of energy because, anytime you participate in research, it takes time and effort. So please do consider it.
Thank you so much. Thank you. Okay. Bye-bye.